Thank you everyone for joining us. We are so excited to have our UC Davis team here with us. Curtis Carlson, who's doing his PhD, Christina Tavilak, Joanna Chu, and Levin, I think, are you there, Levin? Um, Levin Chen, who's helping out the team. Um, this is a really exciting time for us in the SKS world. Oh, we also have Catherine DeMott here, who's actually from Adelia Therapeutics, who's been helping us with our science strategy. So welcome, Catherine. We are super excited to be talking to the community about this today. Joanna and her team have been working with Andrew Liu for the last couple of years around creating SKS in flies to help with Andrew Liu's research and SKS in mice. And we're at a very exciting inflection point where we can start to look at testing treatments, drugs on these flies and see whether we get any hits. It's first for us in the SKS world, and we're really excited to share it with our community. So the way that we're going to do this is to assign maybe 20, 30 minutes for the team to do a presentation to talk about the work that they're doing. As you think about questions, please feel free to put them in the chat, and then we'll use the rest of the time to ask questions. You can feel free to come off mute after the presentation is done to ask a question, or we'll ask them out of the chat. But with that, I will hand over to Joanna.
All right. Hi, everyone. So thank you for being here and giving us some time to let you guys know what we've been up to. So today, Curtis and Christine are going to present on behalf of the team. And I think Susan mentioned Levin. It's a new undergrad who just started working with us about 2, 3 months ago, and he's been really great to help out with the fly work. So I'll let them take over. And like Susan was saying, if you have any questions, feel free to ask at the end. You can also ask in the middle if there are anything that you don't understand or if Curtis and Christine are going too fast. Sometimes when they're nervous, when they present, they go faster. Feel free to, interrupt and ask questions. Okay, go ahead. You guys, Curtis, I think you're first. Yeah.
All right, everybody can hear me? Okay. First of all, yeah, thank you, Susan and the SKS Foundation for inviting us to tell you all a little bit about some of the exciting stuff we've been doing, sort of, what are the next steps that we see moving forward with this project. So today we'll be showing you part of our project studying Smith-Kingsmore syndrome in fruit flies, working towards finding treatments for SKS. So as many of you are probably aware, there's currently no FDA-approved treatments for SKS, and really very little is still known about the underlying pathophysiology of SKS, what is causing SKS symptoms and everything. So our overall goal for this project is to develop fly models for SKS that can be used to study the molecular mechanisms that are underlying SKS, and which will be ultimately used in a drug repurposing screen to hopefully find drug candidates that might be able to relieve symptoms of SKS and improve quality of life for patients as well as caregivers. Our pipeline begins with fly model generation. We could do this through a couple different ways, which I'll touch on in a few slides here, where we actually introduce SKS variants into fruit flies. We then take those flies and do some behavioral characterization to see are these flies sort of mimicking, recapitulating SKS symptoms. Those flies, after being behaviorally characterized, will actually be put into our drug screening assays, where we can screen through different compounds to hopefully see, do any of those drugs, can they alleviate some of those behavioral symptoms that the flies are showing? And in parallel, we're also doing molecular characterization of these SKS fly models to really start to like peek under the hood and see what's going on in SKS cells that's really causing and manifesting into SKS symptoms. And results from that can actually inform a lot of our drug screening assays. And so we've actually been spending the last 2 or so years optimizing all of these steps. It's taken a lot of work going from beginning all the way up to the drug screening. but we feel like we've made a lot of progress through the optimization. And so we feel like we can present to you sort of our best case scenario timeline for really moving forward with the drug screening. So if we look at the bottom of the slide here, we have a sort of timeline we're presenting, and this is per SKS variant that we might hope to model. Take anywhere between 2 to 6 months or so for molecular characterization. Really depends on exactly what we are doing and whether we need to develop additional reagents or even perform even further optimization of some of those molecular assays. And then ultimately, we predict about 3 months or so per variant for drug screening. Really also depends on the number of compounds that are being screened here. And so for those of you who have never heard a talk from us before, you might really be wondering, what does a fruit fly, this tiny fly you see flying above like bananas on your countertop or something, what does that have to do with my child or my family member who has SKS? And what makes flies, fruit flies, a really appealing model for studying SKS? Well, fruit flies have a rapid generation time. It takes about 2 weeks to go from egg to adult. And that makes them a very economical and high throughput model. We can do experiments relatively cheaply and relatively quickly compared to other systems. Flies have a diverse array of genetic tools available for studying things in them. We could pretty much manipulate all aspects of their genome and the genetics and really understand and kind of look at what are the consequences of that. They have analogous anatomy to humans. So despite having 6 legs and 2 wings and an exoskeleton, flies actually have organs and organ systems that are doing pretty much the same thing in a fly as they're doing in us humans. And flies actually have a very rich history of being used in circadian and sleep research. And flies actually do sleep, and they also sleep at night. A lot of people don't think about it, but flies do indeed need their sleep. And perhaps most important to this project, this SKS project, is that the role of mTOR, this is the gene implicated in causing SKS, the role of that gene in development and metabolism is conserved between fruit flies, I'll sometimes say drosophila, I'm talking about the same thing, between flies and humans. So mTOR in flies is doing more or less the same thing as it's doing in humans. And this list of SKS variants or mutations, it keeps growing. Every time we check, there's some more. My last tally, there's about 52 SKS mutations, and remarkably, 42 of those we're actually able to model in the fly. So coming back to our timeline and comparing this against, say, a mouse model, just to really show you the sort of speed that we could potentially be working with here. While flies take about 2 to 3 months to generate that model, a mouse model could take anywhere between 1 to 2 years. Again, best case scenario. Mice take significantly longer to do any sort of behavioral characterization. Typically, they require some training period, and oftentimes you have to wait for the particular age of the mouse to do something. Again, molecular characterization could take anywhere from 3 months to 2 years with mice. And there's simply no way to use a mouse model in any sort of high throughput drug screening. Simply unfeasible. That's not to discount mouse models. They're very, very important tools. But this is more so to highlight that our fly models can actually be a tool towards the next step of mouse studies to potentially inform and streamline those studies. And rare disease studies are challenging due to the time and cost of developing those vertebrate models. And as I said, the fly offers a robust and rapid platform for studying diseases. Remarkably, about 75% of disease-related genes in humans actually have a fly version. And flies have already been used to model many human diseases from neurodegeneration to intestinal dysfunction and inflammation. So they have a established history of being disease models for human diseases. So we'll start here with our fly model generation. And as I said, this list of SKS variants, continues to grow as the community learns more. What we have here is a schematic of the mTOR protein itself, divided up into functional domains of that protein. Basically just means parts of the protein that do different jobs for that protein. And so you can see we have in boxes where some of those variants are located, which domains do they work with. And in red on the schematic, these are flies that we have generated or are in the process of generating that are sort of our germline SKS models. So these are CRISPR-Cas9 edited flies where every cell in the fly carries an SKS variant. Those in blue on this figure are what we are calling sort of mosaic SKS, and this is sort of tissue-specific SKS, where we use a different version of transgenic flies, and we can actually isolate SKS expression to specific cells or tissues to study what's going on in isolation in those organs or cells. And on this figure too, anything that's sort of in italics and gray, those are those 10 or so variants that we're unable to model in the fly. And after we get our fly models, again, as I was saying, we put them through some behavioral characterizations to see if these flies sort of phenocopy or mimic patient symptoms. And we've done that through a variety of ways, and I'll just show a few here, where our flies actually have sleeping symptoms. So I'll walk you through this graph real quick. This is called a sleep profile. On the y-axis, we have time spent asleep. So all the way at the top, that means they're 100% asleep. All the way at the bottom means they're 100% awake. And on the x-axis, we have time and hours split between daytime and nighttime. And so this gray line here is what a sort of healthy control fly sleeps, where they have periods of sleep during the day and a consolidated period of sleep at night. But when we look at our SKS flies, here shown in the red line, you can see that these flies actually have difficulty sleeping during the day as well as at night. They tend to actually start waking up significantly earlier than the healthy controls. Our flies also have seizures or seizure-like behavior. This is actually, I'll wait for this to restart here, but this is actually individually tracking flies. So I'll bring your attention down here once this resets to the lower left corner. You'll see a fly in red once this restarts. And right there, that fly in that bottom corner, that fly is actually having a moment of paralysis, followed by spasms. These are like seizure-like events, and then a period of recovery when it starts like recovers from that seizure. And if we actually quantify all of this, we could see here with this little graph on the side here that our SKS flies have significantly more spasm events or more seizure-like events. Our flies are also hyperphagic or tend to overeat. This is another very simple assay we have where we actually just have liquid food in these little capillaries, and we literally just measure the volume of food that the flies consume over a certain period of time. And in fact, our SKS flies are eating significantly more food compared to control flies. And then finally down here, we have body temperature dysregulation. So our flies actually also struggle to regulate their internal body temperature. So despite being cold-blooded animals, flies still do regulate their body temperatures by positioning themselves in the environment at ideal temperatures to sort of equilibrate their body temperature. And so this sort of assay, we put flies into an arena that has a hot side and a cold side, and they're free to choose where they want to regulate their body temperature. And so the black and gray lines are our healthy control flies, and they tend to stick, not too far away from the hot side of the arena, whereas the SKS flies in red, they tend to move pretty far away from the hot. They stay in the cold, and this is to help them regulate their body temperature. So they're actually running internally hot, so they seek the cold.
Curtis, could I just say something super quickly there, just for the
Yes.
people on the call? Like this is one slide, but it's actually a massive deal for SKS to have like animal models that actually behave like our SKS people behave. It's taken
Yes.
a lot of work to get it to this point, but like a lot of diseases don't even have an animal model that replicates how the condition works. So for us to be in this spot where we don't just have like one symptom of SKS represented in these flies, but they have trouble sleeping, they have seizures, which is a really big deal. A lot of diseases can't find an animal model where they can actually like look at seizure events and even things that all of us talk about with our kids that are hard to like quantify with doctors and tell them about, like this overeating, or like my kid doesn't want to wear a coat in the middle of winter. They seem to run hot all of the time. Like we now have flies that have those behaviors. So as we think about the research we can do, not just, it'd be great if we could find some hits for the treatment, but this will really help so much of the science going into the future to like, how does SKS work? How can we do more research on flies to help us sort of get underneath how SKS works? So I just wanted to put it out there that this is one slide, but it's actually a pretty big scientific deal for us to have an animal model that shows all of these things.
Yes, exactly. This one slide really could be 4 separate presentations in itself. But yeah, it's remarkable. Um, and this is only some of the behaviors that we've seen. So this is great that these flies are pretty much recapitulating SKS, many of the symptoms. But all of these assays are not particularly high throughput assays. They'd be very difficult to sort of expand to screen dozens or more compounds of drugs to see can they rescue that. And so actually, Christine's going to take over the next couple slides, and she's going to tell you a little bit more about other behavioral assays that we're using that could potentially, or we think will be used in our drug repurposing screens.
One really
And
quick question, maybe for Curtis and Christine and Joanna. Is this just the one mutation that you've seen these different behaviors in, or have you seen have you done sort of observed this work in different variants of SKS?
Jana or me?
So I think we haven't ran through all the variants using all the tests, but we have tested I think all the ones with sleep, and so far I think they're showing to varying degree like sleep phenotypes. Yeah.
That's correct.
Yeah, we haven't tested, I think, seizures and hyperphagia. Body temperature dysregulation, I think Curtis is testing them in the new. We have to have access to the platform that we're testing it, and we're kind of waiting in line for that. Yeah. Our collaborators have that in the next building.
But we definitely have, so just to also to Martin, for your question, we have the potential to look at different variants in this way. So that's one of the great things about flies, is that a lot of our different variants are represented in flies. There are a few that aren't, but we actually do find that we can represent those in mice. So one of the nice things that we can do, flies are much less expensive than mice, so we can look at some potential options. And then if we want to test some of the variants that occur in like SKS variants that you can get in mice but not in flies, we can kind of narrow down that testing in mice, which is significantly cheaper and shorter than doing that from scratch with mice. So thanks.
Yes.
I think the other thing that maybe Curtis you want to mention in how we chose, to generate which variants that, what was the basis of the choices? I think we mentioned that, but go ahead. You want to answer
Yeah,
that?
yeah, yeah. Yeah, so our initial plan right now sort of is to try to choose one variant per functional domain of mTOR. Um, there's a handful of domains in that mTOR protein, and there's mutations pretty much in any of them. So the idea there is we'll model one per domain with the idea that mutations within those domains are probably resulting in similar outcomes. So a mutation in that fat domain probably has similar effects, within that domain. Whereas a mutation within the heat domain, those mutations probably have similar effects with each other. So we can first choose one per domain, and then eventually from there potentially expand outward with contingent upon like funding and that sort of thing.
I think
So with
Christine,
that,
Okay,
oh, yeah.
Christine, I don't know if you can control.
maybe Curtis can help you.
Oh, she's controlling my screen.
Okay, there it is. Slight delay. Okay. So as Curtis mentioned, in order to have an effective drug repurposing screen, we need to have a platform that is high throughput, which will enable us to test many compounds at the same time. So if we want to look at several different FDA-approved drugs, we would need to be able to test them simultaneously so we can move faster towards finding an effective treatment. So thus far, the behaviors that we have shown so far and the assays to test those behaviors are not really good at testing in high numbers. So it's very difficult to scale upwards and to do that in a high throughput manner. So we tested 3 additional behavior and physiological assays that are high throughput. And in these assays, our SKS models do recapitulate a lot of the patient symptoms and physiology, as we had mentioned earlier. So these 3 assays are the cold shock recovery assay, a metabolic resilience assay, as well as a climbing assay. And in all 3 of these platforms, we're essentially measuring the fly's ability to adapt to and to recover from environmental stressors or changes in their environment. So in the case of the cold shock recovery assay, we take a vial of flies and we submerge it in an ice water bath for 1 minute. For 1 minute, and during that time, these flies will experience a cold shock, causing them to become knocked out and to fall asleep. After 1 minute, we remove the fly vial from the ice and we put it back at room temperature. And we assess how long it takes for the flies to actually recover from the cold shock. And what we find is that our SKS models actually take longer to recover from the cold shock as compared to our control flies. This shows us that they're having difficulty regulating their body temperature, which is similar to the behavioral assay that Curtis just showed with the body temperature regulation. So as John is mentioning in the chat, they do have difficulty maintaining homeostasis. So the next assay that we tried is called a metabolic resilience assay. And this assay essentially is measuring the fly's ability to deal with some sort of metabolic challenge. In this case, we're doing nutrient deficiency. So we take flies and we put them in a nutrient deficient media, and we monitor how long they can persist within that media. So on the y-axis here, we have how many of the flies were able to persist over a set amount of time within this media. And then on the x-axis, we have the time. And we see that the SKS models actually have a much harder time being resilient to this nutrient deficient media as compared to our control flies. And so we've seen in our in Andrew's human cell data that SKS is a result of hyperactive mTOR. So because we know that mTOR is a master of metabolism and nutrient intake, these flies require much more nutrients, which is what makes it difficult for them to be resilient in these conditions. And this manifests as the symptom of hyperphagia. The last assay that we tested to use for the drug screening platform is a climbing assay. So in this particular assay, flies are placed in an empty vial and we tap them down in order to give them a mechanical perturbation that causes them to fall to the bottom of the vial. And then after a set amount of time, we count how many flies are able to recover from that mechanical perturbation to make it to the top of the vial. And so we've decided to use this climbing assay to move forward with the drug screen. And the reason we chose to use it is because, I'm sorry, when I click the advancement, it's a slightly delayed, and so I'm pushing it twice. My apologies. So with our current setup, we can actually record up to 20 different conditions at one time. So then we have the ability to test many more of these FDA-approved drugs or compounds simultaneously. There is a clear difference between our control and our SKS models, and I'll show you guys that in the next slide. But this is important because when we treat the flies with the drug, it's not always going to be, depending on the concentration of the drug, it's not always going to restore it to that control condition phenotype. So we need to be able to see if even there's a slight change in the phenotype, could it kind of rescue it a little bit? So having that clear difference is really important here. The climbing assay is also very well established in the field of people who use flies in order to do these drug repurposing screens, and they have been shown to be very effective and successful.
So if I could just jump in there too, Christine, just sort of for the folks, the first step of this team was to show that flies sort of have SKS, right? They have the symptoms that SKS folks have. What we're asking them to do now, or hoping to do, is for them to test hundreds or even thousands of different compounds to see if any of those might help with these symptoms. So there's 1,400, 1,500 drugs that are approved to use in the US and across different parts of the world that we don't know whether they might work on SKS or not. Doctors can't prescribe them necessarily to us. They wouldn't think to prescribe them. So the goal is to test those on the SKS flies. But to do that, we need a relatively quick way of doing it, because to test, just looking at the seizures or just looking at the sleep, it would take ages for one fly to do. So these tests that Christine was talking about are a much faster way to show whether the SKS flies behave differently from regular flies. And you can test lots of compounds much faster. And then we can think about what other research we might need to show to see whether it will have impact on different symptoms. But the first step is to actually have a test. So this also took a ton of work, right, to look at what sort of tests can we do to do hundreds or thousands of compounds to actually test them on all the flies.
Thank you so much for that, Susan. As Susan mentioned, we really want a platform that we could use in order to test many of these drugs. And so this particular climbing assay is great because not only does it, in this assay, do we see that they exhibit an SKS-like symptom, we can actually do several compounds at once. And so naturally, flies have the tendency to want to climb upwards when they're in a vial like this. And so if we were to tap the flies down and we give them like a few seconds to climb up, majority of them are going to end up at the top of the vial. And then only very few will actually fail to make it to the top. So in this control fly here, we only have 2 flies that were not able to climb to the top, whereas the rest of the flies in the vial successfully made it. However, in our SKS models, under the same conditions of being tapped down and given the same amount of time to climb up, only half of the flies were actually able to successfully make it to the top, while the other half failed to climb. So you can see the 5 flies down here were not able to reach the top. And this could be for numerous reasons. Sometimes the tapping down can actually induce a seizure. So it's not giving them enough time to actually climb to the top. They could also have muscular and motor deficiencies, which prevent them from actually climbing very far, or as they're climbing, they can't hold on for long periods of time, so they tend to fall to the bottom of the vial. So this is the clear difference in phenotype that I mentioned earlier, where you can just with the naked eye see what the control fly looks like versus what the SKS model looks like. So if we were to administer drugs to the flies, we would hope to restore it to this control condition. So now that we've shown that the assay works, in which you can see this clear phenotype, we're making a few tweaks and adjustments to the assay in order to increase the likelihood that we find hits that are relevant and actually work well for treating SKS. So some factors that we are considering and optimizing at the moment include looking at different ages of flies. So we want to make sure that all of our flies across our experiment are age matched. Also, there could be an optimum age in which a compound can work, depending on what their metabolism and growth is like. So we're trying different ages to see what would be the best one to use for our assay. We also know that there can be differences between males and females. So we're running this assay with males and females separated at the moment to see whether one sex shows a clearer phenotype than the other one. And then we would choose the one with the clearer phenotype to proceed with. Again, because we're trying to increase the likelihood that we find a hit. Other things to consider are what time of the day we're actually conducting our experiments. This is important because as you saw from Curtis's sleep data, flies are more active at certain parts of the day and they're asleep during other times. So if we were to do the assay at a time in which they're not very active, then we might not get as clear of a phenotype, or the hits that we get might not be very telling. So that's actually something that Levin's doing, is he's trying to do this assay at different times of the day to find the best time in which we have a clearer difference between our controls and our SKS models. The final thing that we're doing is we're actually working on dosage concentration, as well as how we administer the drug to the flies. And we're using a known mTOR inhibitor, which is rapamycin, also known as sirolimus or Rapamune. And I don't have the data on this slide. However, when I do administer the rapamycin to the flies, I actually can restore the phenotype so it looks just like the control. So majority of the flies are at the top of the vial and only 2 are actually at the bottom. And that's true for the one in which we fed the rapamycin. So once we can optimize all of these different conditions, then we can actually proceed with the drug screen. And we just got some compounds from the National Institute of Health. So those are ready to go for us once the assay is fully optimized. Okay. So our overall goal here really is to find the most effective treatment for SKS. So the drug repurposing screen is really just the first step in getting to that, in order to get to that step. And so what the drug repurposing screen will do is it will narrow down the amount of compounds that might have an effect on working with SKS symptoms. And then we can use those hits and see whether they actually work for other symptoms as well. So we can run them through all of the other behavioral assays that we showed that are a little bit more labor intensive than this climbing assay. Additionally, we can use these hits to test with other SKS variants. So we know that there's no clear distinction now between which variant causes which symptom or how severe that symptom is. So then we can test many different variants to see how they respond to each of these drug hits. These drug hits could also inform us as well as clinicians as to which bodily processes and pathways are involved in system manifestation. And so then if there's a possibility that maybe we didn't test certain drugs, but if a clinician saw this data, they can find other drugs on the market that could work for alleviating SKS symptoms. And then finally, this information can also help clinicians kind of improve drug delivery as well as management. So let's say, for instance, that there is a certain drug that targets a protein that's only in the body at a certain time of the day. We want to make sure that they're taking the drug when that protein is actually in the body, and so it's effective. So this is a branch of medicine known as chronomedicine. So it's really focused on optimizing the timing of medical treatment to align with the body's circadian rhythm, or our internal body clock. And so it's involved in adjusting the timing of the drug administration in order to increase the effectiveness and then reduce the side effects that could come with taking the drug. And so it's possible that there is a combination of medicine and lifestyle changes, such as particular diets, that could also help with some management. And then Curtis will kind of expand on that here.
Yeah, so moving from that drug screening into a little bit of our molecular characterization. So again, as I said, we're using our fly models to really understand and characterize the molecular mechanisms that are underlying SKS that ultimately manifest into the symptoms. And we're really doing this in a variety of ways. But for the sake of time and simplicity, I'll just show a really simplified outline of some of the stuff that we're doing. So in collaboration with Dr. Andrew Liu, who I'm sure some of you know or have met, we've performed and analyzed proteomics data from human SKS cells. And proteomics is a technique that really allows us to take like a big snapshot of the full protein landscape of a cell. And this really helps us to identify protein pathways that may be disrupted or otherwise dysregulated in SKS. That could be like energy metabolism pathways, cell growth pathways, stress response pathways, that sort of thing. And so we can actually use that proteomics data to really narrow down some of the compounds we're actually screening to really actually get to more targeted drug screening. If we know that there's something wrong with energy metabolism, we could potentially already, narrow down to, okay, we have these compounds that we know act on this pathway. And so we can actually administer those to the flies to see, does it rescue any of those phenotypes, such as that climbing assay that Christine showed. And then on another thing that we're doing with the characterization is we're really trying to characterize the activity of these SKS variants. How are they affecting mTOR's activity in the body, in cells, in the brain, and so on. And so we're actually developing and optimizing assays that can measure the activity of these mTOR proteins. And so here I have a simple graph that we're actually measuring the brain activity, mTOR complex 1 brain activity in healthy control flies. So on the y-axis here, we have the activity of mTOR, and on the x-axis, we actually have time in hours. And so actually what we've done is we've taken our flies at different times of the day and actually measured what's going on in the brain. And we could see that mTOR actually oscillates in its activity in a healthy control fly, where you have periods of low activity around maybe this like 10 hours, where mTOR is kind of low and not doing too much of its job. Whereas up into the late night, we have sort of a peak of mTOR's activity. And so this really highlights and reinforces some of the things that Christine was just talking about, where we can use this sort of assay to really characterize different SKS variants and their impact on mTOR activity. As Christine was saying, there's currently really no clear relationship between SKS variants and how those manifest into symptoms or severity of symptoms. This also can inform us on drug dosages potentially to be used, where maybe one variant produces a very, very hyperactive mTOR. Potentially other variants might be a lower activity of mTOR or could shift sort of the window of when mTOR's activity is peaking or at the trough. And that again also really informs time of drug delivery. Do you want to administer a drug when mTOR's activity is very low in this variant or very high in this variant to sort of normalize to a healthy level of mTOR? So again, coming into that chronomedicine that Christine was talking about, time of day of drug delivery can be very important. And so moving forward with some of our future directions, things that we're thinking about or working towards right now is expanding our patient symptom characterization of our fly models. This isn't one of our SKS flies, but this is an example from, I believe, an autism spectrum disorder model, where we can actually record sort of stimming behavior in flies, repetitive motions. This fly is like doing this repetitive grooming behavior over and over and over. We could also expand to other behaviors such as aggression, where we can actually record and score different aggressive behaviors in flies Let's see, do our SKS variants show increased aggression or increased stimming behaviors? We are continuing to work on our molecular studies to really understand how SKS is perturbing different signaling pathways in the body. And again, going back to what I just showed you in the previous slide of measuring SKS variants impact on mTOR activity, a very important thing to do moving forward. And then again, as a big focus on this talk is really pushing forward with our drug screening in the FLY platform to hopefully find some potential compounds that rescue some of the symptoms in the fly and move those towards the next stage of potentially finding treatments for SKS symptoms. So with that, I'd just like to thank everybody for joining us on this call and to really thank everybody in the lab who is a big part of this project. Obviously, Joanna Chu for really running and running this project and taking us all on. We have postdocs that are great at this, like Christine and some others that have joined the lab. We have a great team of awesome undergrads that are part of this research. Everybody here in bold have directly contributed to this project in some form. And we have a lot of past members that have contributed to this. Some of you might have met Gary Chow in the past. He was a postdoc who really laid a lot of foundations for this project and got the ball rolling. And I'd also really like to thank Andrew Liu, our collaborator, who's working on mouse models and some human cell data for this, as well as our funding from the NIH and everybody at the Smith-Kingsmore Syndrome Foundation. So with that, we'd be happy to take any questions.
Thank you so much, Curtis and Christine and Joanna and Niven. Really appreciate it. Maybe, Curtis, you can pull down the screen so we can see the gallery view of folks. Yes. This is really exciting work. I think, one thing I will say, we've been working with this team for the last couple of years. Your contributions have helped fund this science. And, one of the things I will say about this team is they listen so much to the patient experience. So, we talk to them about, look, we've got random stuff. Our kids are hot all the time. Our kids eat a lot and they've really listened to us. Christine, Curtis and Joanna came to our conference, met with families, heard a lot about our experiences. And so I think we are most definitely trying to find hits, trying to find treatments that could help, but they're really thinking about it long term in terms of what science can we do to help with things like timing of dosages. For anyone here who's given their kid a big dose of melatonin too late and had the kid wake up 3 hours later having had super sleep to destroy the house, will understand we all kind of like instinctively know that timing probably is going to be a big factor with us. Now we're helping build the models that we can hopefully get better answers on these things. So I would like to thank the team so much on behalf of the community for really partnering with us and listening to us and knowing what matters to us. And with that, if we've got any folks with a question, we'll welcome you to come off mute. I do have a lovely comment from Shiloh and Melanie. Thanks, Melanie, that Shiloh is one of our SKS community and he doesn't have a question right now, but he loves what you're doing and you're heroes to him. So thanks, Shiloh. We appreciate that. Anyone have a question? Like that first day of school, asking for questions.
Jack doesn't have a question, he says, but he's been very interested in this entire presentation. That's right. He's been very interested. Yeah.
I mean, I have a question. So we talked earlier in the presentation. So there are different sort of like groups of mTOR, different domains of mTOR, and we're going to be looking at variant, like sort of a variant from each one to see if it's different. So far, we don't see much kind of correlation. Like we don't, folks in one domain don't have dramatically different symptoms from those in another. And sometimes people with the same variant have really different symptoms, even with exactly the same variant. So Do you think that's something that we can look at? Like, say, if you've got the same variant but different symptoms in humans, is that something where you think that maybe there could be a timing issue, or male and females might look different, or something that you could be able to test for with the flies?
Yeah, I think definitely. I think environmental factors definitely come into play. Interesting you asked because I remember last time when we went to meet you guys in Chicago at the conference, we asked you about diet, whether you guys keep a diet record. So Curtis has actually started testing different dietary conditions. Like we can feed our flies different food, which is pretty easy, and then see if it modulates some of the sleep phenotypes or other phenotypes. And he actually see that it has a huge effect, at least on sleep symptoms. So when he changes like protein content or sucrose versus glucose and just different percentage of the sugar, he sees actually a big difference. So I don't think it's too surprising that even with the same variant, you get sort of different levels and, different manifestation of the phenotypes. Yeah, this is definitely something that we're interested in. Yeah.
Lauren, are you able to unmute? Oh, looks like you are.
Yeah, sorry, I'm having technical issues. I joined right at the end, sorry. So you guys might have talked about this, but I'm gonna try to say this without crying, but so top of mind for me is seizures. I lost my son in March due to his epilepsy that's drug resistant. So I'm just really curious about just the work on how, I guess, the broader plan is. Like, I know it's long term, but given how we know we have so many kids that are drug resistant, and kind of managing some of these things that are related to SKS, but are kind of like symptoms aside and a whole nother kind of diagnosis we have to treat, how does this work within that?
So, can you repeat your question again? Sorry. So
Yeah, sorry,
longer
I'm probably
term.
not. I don't know if I'm making sense. I guess like just seizures are a big thing, and I think that that's what a majority of, unfortunately, our kids are
Yeah.
passing away from. So Mason had drug-resistant epilepsy, basically, and we tried multiple forms. I mean, he had a fantastic neurologist, was followed and everything, and he it was SUDEP that he passed away from. But I mean, I guess given how on top of I feel like our families are and stuff, and just thinking about all the timing and like, I'm just curious if long term, is this drug going to be something that also will help manage epilepsy and the seizure activity better within our kids?
I mean, I think the
Yeah.
goal would be, Lauren, so seizures is something that we can see in the flies, and knowing that some of our kids do have drug resistant seizures, there's for sure the opportunity to look at those variants and test different drugs on them to also see whether there could be additional things, like whether that's the time of day, amount of dosage. Unfortunately, tragically, it's never going to come soon enough, right, and fast enough. One of the other things that's a bit that with this sort of research is there are a lot of rare diseases that are doing this sort of research with fruit flies, looking at it in different mutations and seizures is top of mind, I would say, for all of them, right? Like it's such a awful, awful burden for everyone. So for us, seizures is top of the list in terms of the symptoms that we would be looking at testing. God willing, we find something that could be helpful, especially for those. But we would, we do have an opportunity to look further into that as part of this work. Joanna, maybe I'll throw to you because you're the actual
Yeah.
scientist.
Right. Yeah. So I think that what we do in fly genetics is this might be useful is, let's say if there is a drug resistant happening, what we can do is if we have a fly line, and then we can actually find that shows a seizure phenotype, then we can actually do a second site we can do a screen for genetic mutations that modulate that seizure phenotype and be able to find specific phenotypes, like specific mutations that could modulate the seizure phenotype. And through that study, we'll be able to see what other drugs and what pathways might actually modulate the seizure phenotype and be able to maybe solve the drug resistant issue. Yeah, so there are definitely methods using the fly genetics to solve that. Yeah.
I think that's really great. And it just made me think of just even having more research to share. Because, like, we were down a path where we were pushing Floor Mason to get a VNS, and they had to present it to the board and all this stuff. And they were too slow because it's just like, I mean, what we're all facing in all rare diseases are just data and information. So I just think for other families to be able to show up and say, like, this is what we know. So, like, we need to do this now before it's too late,
Yeah, definitely.
Thank you. I appreciate it.
Thanks, Lauren. We really appreciate you joining the call.
Thanks, Lauren. There's a question from Trevor Martin is his first name. I think that he goes by Smith on the chat if you guys want to take a look at that one next.
Okay, so I think Trevor, what's the biggest
challenge?
Yeah. Well, the biggest challenge is sometimes the fly is great in terms of genetics and using it for modeling certain behaviors. But, I think one of the challenge is logistics to be able to actually screen through all the different variants. We have to create all the different mutations. I think it's more sort of a little bit more labor intensive if we include more variants, and also the fact that we need to make everything high throughput so that we can screen for all the possibilities. That's great. And one thing that would help is that's one of the reasons why we conducted the proteomic screen to be able to not just test using the high throughput screen platform, but also maybe use the proteomics to find downstream pathways that is modulated by mTOR that actually results in all those symptoms. Once we know what all those pathways are, we'll be able to actually find drugs that are more targeted that we could test in the more labor-intensive assays like sleep, body temperature rhythms, those type of things. So I think that the amount of work is one of the challenge, but I think we're trying to actually develop methods and ways to have some shortcuts to make things a little bit faster. Which drugs are you most excited to test? I think that is also sort of, which I would say that we're less excited about testing drugs that directly modulate mTOR because we feel that mTOR it regulates so many different things that we're worried that it could have side effects, right? But we would still screen for them. But what we're excited about are actually, like I was saying earlier, using the proteomic screen to isolate pathways that target downstream pathways that are regulated by mTOR and find the drugs that actually are targeting those pathways. So those are the ones that we're actually excited about. And then the third question is, what is one thing you haven't had a chance to say that you really like us, the community, to know? Can you think of anything, Curtis and Christine? I think one thing that we haven't said yet is we really appreciate the support for trusting us and for trusting us, because we know that it's hard to be patient and it's hard to wait while we're doing the research. But I think that I speak for Curtis and Levin and Christine and the team that I think they're working really hard to try to get things going and get the drug stream going. But I think it's been hard work the last 2 years, but I think we hope that it's worth it. And then, we're really excited to get to this point now that we have so many different assays, not just high throughput screen for the drug repurposing screen, but also other behavioral assays that we can use to validate the drugs that we find. Curtis and Christine, do you guys have things to say that you would like the community to know that we haven't said yet?
I think the only thing I would like to really say is how important it is to get as much information as possible from all of you about your experiences, about what you see your kid going through, because as scientists we rely on published literature oftentimes, and with rare diseases, there's very little literature to go off of. And so just like I think Susan was saying earlier, or somebody was saying earlier, when we met a lot of you last year in Chicago, we're hearing, little anecdotal things all over the place. Like, that's not reported in the literature. I've not heard that from a clinician or anything. And so all of that information is really, really useful and important. And I know it's a mountain of stuff to go through. But that sort of steady progress of trying to connect variants with phenotypes and severity of those, and how many people are actually impacted and share that is all really, really, really critical information for us, and really helps us move everything forward, with more clarity and everything.
And Valerie actually had a comment about that. So Valerie, about different drugs that have been tried and they've worked or not worked. So Valerie, would welcome. I'll reach out to you. We can connect to get any of the information from you on that. Valerie also asked a question about rapamycin, whether we have a clinical trial on that. There is not a clinical trial at this point on rapamycin. I think one of the big questions with rapamycin is whether it would work much better if we got it in low doses very early to children versus in puberty. But again, this flow work could be something where we drill more into dosages and at different points too. But it's definitely on the list. But to Joanna's point, rapamycin hasn't worked for SKS the way that we might anticipate it working, or the way that it's worked for, say, other diseases. So it's still a bit of an open question. But Joanna, I just saw you come off mute. Maybe I don't know whether you've got thoughts on rapamycin.
I think that I was looking at Melanie's question on there about Curtis's observation on diet changes. Do you want to mention some of your results, or it's too early?
Okay, so it's just a question to see whether you could elaborate a little bit on whether you've seen anything in diet and.
I have one slide. It's not a whole lot of data right now. Still working through a lot of it, but I can share my screen again and show that one slide.
Yeah. The other thing while Curtis was, I remember what I was going to say now. In terms of rapamycin and other drugs per se, so one thing that we can do in flies is we can actually express the mutation, the variant during development, all through development, versus only in after a certain age. So in the future, if we have drug targets that we can, we found from the high throughput screen or more targeted screen, what we can do is we can actually tailor the fly model to only express the variant during development, treat it to see if that helps in terms of the symptoms, or we can actually bypass the developmental stage and then only treat it in the adult stage and see whether the effect is different. So in flies, we actually have the ability to do that and allow the expression of the variant at different stages of life, and then treat them, during development and after development, during the adult stage. So we can actually sort of tease out the effect of the drugs at different developmental stage. So that might help to, help understand the disease a little bit better as well, and also finding like proper therapeutic route. Yeah.
And touching on some of that, how diet can sort of modulate or impact some of these symptoms, such as sleep that we're seeing. Again, if you kind of look at the simplified mTOR signaling pathway, really some of the main things going into the pathway are nutrients, right? Amino acids, energy from sugars or carbohydrates, growth factors, insulin being released. And so it's not too surprising that diet could potentially really impact and modulate the activity of these variants. And so what I've done is varied some of the nutrient content of the food that the flies are eating. You can see here, if I administer the flies 1% glucose, the SKS flies have a really difficult time sleeping. This might not necessarily be part of their sleep drive. It could also be related to like a hunger drive where they're actually sort of in the starved state and they're constantly waking up, with this foraging behavior, looking for more food. Whereas a wild type fly, even under this sort of nutrient stress, is still able to sort of, hunker down and go to sleep normally and pretty much try to wait out until there might be more nutrients available. If I kind of bump it up to 5% glucose, you can see it actually partially rescues that sleep phenotype. These SKS flies they still have some difficulty, but they're much closer to like a healthy wild type control fly. And then likewise, if I keep pushing it the other direction, giving them even more glucose, so even more substrate for mTOR to be activated, or not substrate, but then you start to worsen their sleep phenotype again. So it's kind of this potentially like a seesaw balancing act here, where maybe in conjunction with potential drugs, diet can be used to help modulate some of these SKS symptoms or behaviors. And I don't have it today, but we're also looking at more detailed analysis of these sleep patterns to see. So flies also have different stages of sleep: light sleep, intermediate, and deep sleep. And so we're also looking at when they're sleeping, what type of sleep are they having? When is that type of sleep happening? Are they having deep sleep when a healthy fly is not in deep sleep? Are they constantly just stuck in sort of a very light sleep, and so they're prone to being aroused and woken up and everything? So really getting with like a sort of fine-tooth comb to really dissect some of these symptoms and phenotypes and how the environment is impacting them as well.
Thanks so much, Curtis. That's actually super, super interesting stuff. We did have one question from Eric, which is actually on the topic of funding. So, what can parents do to help? How will it be funded? I think candidly, the more we get funded from the community, the further we can push this research is the simplest answer. So, there's a lot of steps here. Our campaign this month has been around funding this sort of first step of drug repurposing work. But the more that we can, gather our friends and family to make donations, the more that we can sort of fund this team to push the research into different directions. And also another great thing to do is to join the patient registry, so that we, can track the variants that our SKS kiddos have and what their symptoms are is really useful. You can always reach out to us if you have trouble with the registry, or you just want to like sort of give a download on how you're experiencing SKS. But this month you will see on the Facebook page ad nauseum me putting the link to donations. If you want to share that with family and friends, we actually find that fundraising is much more effective when it comes from you requesting folks than strangers requesting. So you can let them know you've been on this call, heard about the exciting science, and include the link to donations. But we're going to keep pushing forward because this is really, really exciting work, and I think super promising to try and find questions for the kids.
Well,
the link to fundraising in the chat as well, just in case anybody is not on Facebook or doesn't see it. There it is.
Well said, Susan. And I just want to say, Susan, Mike, myself, all the members of the SKS Foundation have done a lot of work with other rare disease organizations. And for those in our community, we are really lucky that we have a dedicated team that has been working and walking this journey with us. So Joanna and her team have really been there for the last couple of years. Andrew starting that work off, and it all started with our community fundraising from the beginning with Andrew and leading to this work here. So we have gotten here because of our community, and we will continue to push this research forward because of the dedication that we see from you guys. So we appreciate you so much, and just love the work that you're doing, and we can tell that your hearts are in it. Coming to our conference last year was just incredible to see you. Was it last year or the year before? Just incredible to see you in person and connect with you, and for you to see our children. So thank you so much.
So appreciate it. Got a couple of questions. I'm going to try and wrap up reasonably quickly so that we can kind of stick to the time. A keto diet. Martin asked about the keto diet and flies tolerating it. I think some of our community actually do follow a strict keto diet to help with seizure management, but I don't know whether is glucose the equivalent, Curtis, of a keto diet, or is it we're just more looking for connections with diet right now with symptoms versus anything very specific?
We haven't tested keto diet specifically, but we definitely can. I mean, so my lab actually worked on how metabolism affects circadian rhythm and sleep. So we do a lot of experiments actually using high-fat diet, different nutritional content, and how it regulates circadian rhythm and sleep. So we could easily just do the same experiments in the SKS variants. And that's one of the reasons why actually Curtis started doing some of the experiments that he's talked about, different percentage of glucose, because this is something that we routinely do in the lab. Yeah. So we can definitely do high-fat diet. Yeah. And high-fat diet does a lot. You do all kinds of, all kinds of symptoms for flies, similar to humans. And fly, actually, it's a good obesity model. It's a good, diabetes model. So we can definitely study metabolism and different dietary conditions in flies. Yeah.
That's great. What I really love about this research, and all of us know from loving people with SKS, is that it really does feel like a whole body disorder. We know it's one gene, but everything is off. GI is off, behavior's off, sleep is off, seizures, obviously. And the great thing about this team is that they're really looking at the holistic ways of improving the quality of life of our kids, because we all know better sleep leads to less seizures. Less seizures obviously is going to be a much better life for all of us. But it's super exciting work. And with that, I welcome, if any of you have follow-up questions, please feel free to email me or Sarah or SKS Foundation at sks.org. We can make sure we follow up with those. We will probably have an update with this team fairly soon. We're going to be having a conference again at the end of April where we'll be inviting this team along to talk. So announcements about that coming soon. But it's really amazing work. We're so thankful for this team, and I'm really thankful for everyone who joined us tonight. I hope you got something out of it and are as excited as we are by the promise of this drug repurposing work. So thank you so much. Thank you, Joanna, Curtis, Christine, Levin. Really appreciate it.
Thank you.
Good night, Jack. Good night, everyone.
Yeah, thank you so much.
Thank you.
Thank you.
Good night, everybody.
Bye. Bye, Sarah.
Bye.
Thank you again.